as mentioned, I am Vinolia, and I'm going to talk about using GWAS approach to identify genomic regions that are associated with Laccase production in Ceratocystis albifundus. So it's very important to give you an overview of what Ceratocystis albifundus is. So the genus Ceratocystis includes various economical important pathogens, of which one of them is Ceratocystis albifundus. So Ceratocystis albifundus is a causal agent of Ceratocystis wilds and Acacia mense, of which Ceratocystis wilds are identified by cankers and lesion on the back. In the final stages of disease, wild, dieback, and death, of course, can occur. This then results in low yield and low quality of wood that is produced, then finally affecting negatively on the economy of black wattle plantation. Ceratocystis albifundus has been isolated from a wide host range, including native and non-native hosts. This then was seen that this fungus can be non-pathogenic or pathogenic depending on the host. The Ceratocystis albifundus was then identified to cause discoloration on the sapwood of Acacia mense and later to cause disease on the cultivated Proteus species in the Western Cape province. This was thought to represent a recent host germ. However, Ceratocystis albifundus pathogenicity is unknown. This led to the study of Laccase in Ceratocystis albifundus, as Laccase was involved in pathogenicity in previous studies. So I'll explain what Laccase is in the following slide. Laccase are multi-copper enzymes present in many prokaryotes and eukaryotes. They also play a role in degradation of phenolic compounds such as lignin, and they are common in fungi and have been suggested to participate in host colonization by pathogenic fungi. So below is the few functions that Laccase possesses, such as biosynthesis of lignin, fruit body formation, cuticles, and pathogenicity. So this then led to our aims. The aims of this study was to use GWAS to identify genomic regions that are associated with Laccase production in Ceratocystis albifundus, and then use Agrobacterium mediated transformation to confirm the role of the identified genes, and finally perform inoculation studies to determine if these genes are associated with pathogenicity in Ceratocystis albifundus. So then this will be achieved in the following methodology. So the first part of the methodology is genome-wide association studies, of which the SNPs were identified using golden helix SBS, and we also observed that the genomic composition of the pathogenic and non-pathogenic Ceratocystis albifundus were different. So the fungus that was isolated from the non-native and the native host was stained with alpha-thiophyll for Laccase testing. From these, we saw that the ones that turned purple indicate Laccase activity, and the ones that did not turn any color showed no Laccase activity. And interestingly so is that our results indicate that isolate from the non-native Acacia and the native Proteus species indicate Laccase activity. So the results of this study indicate that Laccase may be involved in the pathogenicity of Ceratocystis albifundus. So we are in the right step. So going forward, we now want to confirm the identified regions that are responsible for Laccase production in part 2 of the methodology. So using GWAS approach, we identified a candidate gene that may be involved in the Laccase production. This gene encodes for multi-copper oxidases that have been shown in yeast to have Laccase activity. So in order to confirm the GWAS result, we generated a knockout construct of this multi-copper oxidase gene. For this, we then used Agrobacterium mediated transformation protocol, which involves the generation of the construct, transforming it into Agrobacterium, and then the transformed Agrobacterium is co-cultivated with the cornidia of the fungus. So on the following slide, I'll be taking you through the steps of how far I've came about with this project. I have generated the construct and confirmed the construct by gel on the right hand side image, of which it's just a duplication of all the plasmid that I was able to extract, and I used the brighter ones, which were plasmid 12, in the following transformation. And then I've also co-cultivated the transformed Agrobacterium with the cornidia of the fungus, and currently I'm waiting to see if I have transformants. So for future work, we'll be characterizing the fungal transformation using single sequence analysis and southern hybridization, and characterizing the transformants based on morphology and the cultural properties, and then the analysis of the Laccase production in the transformants using the staining and biochemical approaches, and then finally applying the transformants in growth studies and virulence assays on Acacia mensis seedlings to confirm the role of the G92 multicopper oxidase FET3, which was our candidate gene in Ceratocystis albifundus. So in conclusion, we were able to identify the regions and the genes that are associated with Laccase protection and that likely influence the pathogenicity and growth rate in Ceratocystis albifundus. So the functional characterization of these genes is needed to confirm the involvement in these important fitness and virulence traits of Ceratocystis albifundus. Our findings will also demonstrate the power of GWAS for unraveling the genetic basis of phenotypic characters in fungal pathogens. This project would not be possible without my supervisors, mentors, and funding bodies and institute. But I'd also like to thank you very much for the opportunity of having me. Thank you.
Okay, thank you, Vinolia. That's quite a big project. I'd like to know, did you do the sample collection as well as the GWAS and all the laboratory work too by yourself?
Thank you for the question, Natasha. I didn't do the GWAS part. My colleagues and my supervisor already did that part. So I'm just, I already did the transformation. So I'm just confirming the GWAS gene regions that were associated with Laccase activity to see if they're responsible for pathogenicity of Ceratocystis albifundus.
Okay, alrighty. Any other questions? Okay, it doesn't seem like it. So thank you very much, Vinolia.